Hey guys, so we're starting the day 6 video today. And today we're going to be talking about a whole bunch of different structures found inside of the cell. The first structure we need to talk about is the Golgi apparatus. And as you guys can see, the Golgi apparatus looks a lot like the endoplasmic reticulum. The only difference is it's further away from the nucleus. So if you're asked, for example, where's the Golgi apparatus and where's the endoplasmic reticulum, just know that the one that is not directly next to the nucleus, this is the Golgi apparatus. You can also shorten it as GA. All right, so the main job of the Golgi apparatus is to modify, sort, and package proteins. All right, so let's go ahead and look at this for a second. All right, so you guys remember that proteins are built in the rough endoplasmic reticulum by the ribosomes, and then they are transported in vesicles to the Golgi apparatus, right? So they leave the rough ER and they move towards the Golgi apparatus. Once they are in this area, they are modified and sorted and turned into active forms of themselves. And then after that, they are going to be transported to other parts of the cell. This is an amazing video. Honestly, you guys should go watch it on your own. All right, but basically the idea here is that the Golgi apparatus takes proteins from the rough endoplasmic reticulum and basically activates them. Sometimes in the rough endoplasmic reticulum, you just get the basic body of the protein, but it needs to be further modified before it can actually be turned on and start doing its job. And then from the Golgi apparatus, that protein, which is now fully activated and ready to go, will be sorted and transported after it's packaged to a different part of the cell. Okay? Now, transportation of materials in cells is done by vesicles. Like you guys saw in the video, inside the life of cells, right? You guys can see that vesicles are formed just from a part of the membrane pinching off, and the material is found inside of the vesicle, which is like a bubble, and it carries it to a different part of the cell. So a vesicle is basically like a transportation unit or a bus or a car that carries certain things to different parts of the cell. All right? And it's just made up of a little layer of membrane which can fuse with other membranes and allow certain things to enter and exit different areas. All right, the next thing we need to talk about is the vacuole. Now the vacuole can be similar to the vesicle. It is also formed from a layer of membrane. However, a vacuole's job is not transportation, it's storage. So if you look inside of a plant cell, plants have a huge central vacuole, and this is a permanent structure inside of their cell. As in, the central vacuole is always there. Sometimes it's full, sometimes it's a little bit less full, but it never truly disappears inside of cells, inside of plant cells, okay? So the large central vacuole inside of plant cells can be used to store food, waste, and water, right? So mainly water in there. Now, animal cells do also have vacuoles. However, our vacuoles are very, very small and they disappear. They're not permanent structures inside of our cells. So they'll store a specific material, just like here, vacuoles are made for storage. They will store a specific material, and then once that material is used, the vacuole will go away and it'll form again when a different material needs to be stored. But they're not permanent structures in animal cells, and this is why if you're asked which type of cell has a vacuole, just say plant cells, because we're assuming that you mean the large central vacuole, not just small and, temporary vacuoles. All right, and the last organelle that we need to talk about is the lysosome. This is a very cool organelle. It's found inside of animal cells, and its job is basically to patrol the cell, to look around and find things that are not supposed to be inside of the cell. So The lysosome will find worn out or broken cell parts, so parts of the cell that are not doing their job properly anymore. Maybe they were damaged by something or they're just worn out because, it will basically eat that part of the cell and release enzymes which will break down that piece into a whole bunch of little parts. So let's say that there was a protein that was no longer doing its job properly. The lysosome would come, it would take it in, and it would use enzymes to digest it into small pieces. And then those small pieces can be used to build other things inside of the cell. Remember, the cell is dynamic, it is constantly changing. If you no longer need a specific part of your cell, the lysosome will come, break it down, and the parts that it broke down will be used to build other things. It's like Legos. You have a structure, you break it down, you can use the Legos to build other things. All right? And another very important thing that the lysosome does is that it helps to digest bacteria and viruses in some cells, right? So certain cells in your immune system will go and they'll find invaders like bacteria and viruses. They'll take them in, then the lysosome will come and it will start to break down those bacteria and viruses. Lysosomes are extremely important and they have multiple roles. You can call them the garbage collection of the cell because they help to find things that are not supposed to be there. They help to break them down so that they can be used to build other things. All right, so just remember, the Golgi apparatus modifies proteins, it sorts them and packages them and sends them to other parts of the cell wherever they're needed. Okay? Vesicles are used for transportation. Vacuoles are used for storage. And the lysosome is the garbage collector of the cell. All right, guys. So go ahead and work on your quiz and good luck.